So chapter 16 is a continuation of our genetics unit. Basically, we've been talking about how genes get passed from generation to generation and the process that it works and the different ways that it does that and what happens if it goes wrong. But now this chapter is about the actual molecules that do that, meaning DNA. And so the very first thing in your textbook is this picture of Watson and Crick, James Watson and Francis Crick, who are the two scientists who discovered the structure of DNA. And you guys are going to have that down without a question, but they are very famous, and we're going to talk a little bit about what they did to get there. So back early in the 20th century, so the early 1900s, scientists were trying to figure out what was going on with these biological molecules that they knew existed, meaning DNA, protein, carbohydrates, and lipids. They knew that the four of them were there and they were important and they were found in all living things, but they didn't know their structure, they didn't know exactly what they did, and so they were trying to figure all this stuff out. They didn't have nearly the same technology that we do today where it's super easy to figure out those sorts of things, so they had to be really creative and figure out exactly what was going on. So remember Morgan's experiments with the fruit flies showed that DNA is, or the genes are located on chromosomes. And so then they knew what chromosomes were made up of. They knew they were made up of DNA and protein because they could tell when the molecules were present, but they didn't really know what they did. And so it basically came down to those two things. Is it DNA or is it protein that causes, or that is in charge of passing on genes? So people started to do a lot of experiments with these things, and it became really important to choose the right sorts of organisms with which to do these experiments. And so a lot of the early research was done on viruses and bacteria, which was really great because they're so simple. Think about a human body and how many cells it's made up of, or a rat, or a mouse, or a rabbit, or anything that they might have been studying, and how many cells are involved in that, whereas like a bacteria is one cell, and it's good for basically the basic functions of life. It's a prokaryotic cell. Or even a virus, which we haven't really talked about yet, but viruses are not alive, they're not cells, they don't have organelles, they just have DNA and protein, or RNA and protein. So it was really important to start with these simple things, that way they could get the basics down. And so the first experiment that was done that led us to believe that DNA is the genetic material was by a scientist named Frederick Griffith. And you guys learned about this in intro bio. But he was looking at a strain of bacteria that causes pneumonia. One that did cause pneumonia and one that was harmless. It couldn't give you pneumonia. And this picture is on page 307 in your textbook. I lie, 306. And basically, he had 4 different setups. He would take the living cells and he called them R cells, and he injected them into a mouse and the mouse would die of pneumonia. And then he took the living R cells, and these were the cells, these stand for smooth and rough, smooth and rough. And so he took the living rough cells, and if he injected them into a mouse, the mouse lived. So these guys are not, the cells are alive, but they're not causing pneumonia. And then he discovered that if he took the smooth cells, these guys are pathogenic, meaning they cause pneumonia. He takes them, he injects them into the mouse doesn't die because he killed the cells first. He heated them and boiled them. So that's why you boil or cook chicken for a long time before you eat it to kill all the bacteria in it. Different story. And so the heat killed S cells. The mouse was alive. And then what he did was he took the heat-killed S cells, so the S cells caused pneumonia. He killed them, so they wouldn't cause pneumonia. He mixed them with rough cells that were still alive. So 2 things that don't cause pneumonia. He took the living R cells and the dead S cells, combined them together. These guys shouldn't cause pneumonia because neither of them caused pneumonia on their own, but when he combined them, the mouse died. So some living cells, so what he was able to determine was when he combined the living cells that were non-pathogenic with the cells that were dead but pathogenic, the living cells became transformed into pathogenic cells. Basically, they were not harmful, and they got mixed in with the dead harmful cells, and then they themselves became harmful. And Griffith called this phenomenon transformation because he was transforming these non-harmful cells into cells that could cause pneumonia. and it's defined as a change in genotype and phenotype due to assimilation of foreign DNA. So it's not just a change in phenotype. The cells didn't just learn how to be dangerous. They actually changed their genotype. They picked up the DNA, now have that DNA to cause pneumonia, and so their phenotype changed also. And so Avery, McCarthy, and McLeod did another experiment, and they basically confirmed Got it. Avery, McLeod, and McCarthy did a kind of a follow-up to Griffith's experiment where they took DNA, RNA, and protein and saw which one was causing the transformation. And they realized that it was only DNA that caused the transformation. And so it must have been DNA that was carrying the genetic material. But mostly people didn't believe them because there was so little known about DNA that they didn't really think that it would be possible. And a lot of people just thought that protein was the genetic material because it was so complex and there were so many different kinds of proteins and it could do so many jobs. It just made sense that protein would be the genetic material. And then they did, there were studies using these things called bacteriophages. So a phage is a virus, and a bacteriophage is a virus that infects bacteria. So there's viruses that infect our cells, like a cold or like HIV, and there's cells that or viruses that only infect bacteria, and so that would be a bacteriophage. And so this is a picture of a bacteriophage infecting a bacterial cell. Basically, they are just made up of protein and DNA. So they have this protein coat and these little legs that they attach to things with, and then they inject their DNA into a bacterial cell, but only a bacterial cell. This is a bacteriophage. So this up here is made of protein, and they store their DNA inside of here. They land on a bacteria, and they inject their DNA into it. So that's what a bacteriophage is. So then Hershey and Chase did an experiment using a bacteriophage, little guy, known as T2. And what he did was, they knew that this T2 was only made up of protein and DNA. So, and they could see via these pictures right here, probably not this picture, but something similar, that something was leaving the bacteriophage and going into the bacteria during infection. And so they were like, okay, it's either got to be protein or DNA because that's the only two things inside of this bacteriophage. So what they did was they did two different setups. They took the bacteriophage and they basically colored the DNA in one experiment and colored the protein in the other. So in the first one, they made the protein radioactive, so it's lit up pink. And the second one, they made the DNA radioactive, so it's lit up blue. And then they mixed them in with the bacteria, and something was injected into both of them. And in the ones where they injected the protein, or colored the protein, the bacterial cells were not lit up. So there had been no colored protein injected into the bacteria, whereas in the ones where they colored the DNA, I know you can't see this in this picture, but it's on page 307, the remnants, the viruses that were left over at the end, were not colored, but the bacterial cell itself was. So that means that in this setup down here, the viruses had injected their DNA into the bacteria and not their protein. So the protein remained outside of the bacterial cells up here, and the DNA was found inside of the bacterial cells down here. So that was Hershey and Chase, and that basically concluded that DNA was the genetic material and not protein. Another experiment, so there's lots of experiments in this chapter, is by a man named Erwin Chargaff, and so it was already known that DNA was a polymer, meaning a large molecule made up of a lot of the same tiny repeating units, poly meaning many, and They knew that the polymer or the monomer was nucleotides and that they were made up of a base, so that's A, G, C, or T, a sugar, ribose or deoxyribose, and a phosphate group. So they knew that. And Erwin Chargaff was able to determine that the kind of DNA was different between different species, which was really interesting. So, for example, he saw that in humans, 30.4% of the base pairs in DNA were are adenine, but only in E. coli, the bacteria, was only 24%. So you notice that it was different between living species. And so, and the molecular diversity between living things led people to think that this could be DNA, right? Because our DNA isn't going to obviously be exactly the same as a bacteria because we're just a little bit different than bacteria, right? So it makes sense that there would be a difference in our DNA structure between bacteria and humans. And there's two rules that became known as Chargaff's rules, and you'll need to know these. And the first is that the base composition of DNA varies between species. So our base pairs are going to be really different than a monkey's, are going to be really different than a banana's, are going to be really different than a snail's, and really different than a bacteria. So that's really important. The second one was the ratios of certain bases will always equal each other. So in any species, it doesn't matter which one, the number of A's will always match the number of T's, and the number of C's will always match the number of G's. So A's won't match C's, G's won't match T's, but A will always equal T, C will always equal G. And Chargaff was able to determine that, and people thought it was cool, but they didn't really understand what that meant until the structure of DNA was discovered. But Chargaff was able to determine those two things, that the composition of DNA varies between species, and that the number of A and number of T are equal, and number of C and G are equal. So after all those experiments, we were finally able to see that it was DNA that's the genetic material, not protein. And now people are trying to figure out the structure of it, because we know that it's it, but we don't understand how the structure has anything to do with it. So there are these two scientists named Maurice Wilkins and Rosalind Franklin, and they were using X-ray crystallography, which is a very special kind of X-rays that I don't understand, and they were looking at molecular structure. And Rosalind Franklin took a very famous picture of DNA, this one right here, that looks like, that was the basis for understanding DNA, basically. There's Rosalind Franklin. And so these pictures were given to Watson and Crick, or a lot of people saw them, and Watson and Crick looked at it, and were basically, something like that day, came up with the structure of DNA. And so they were able to deduce that DNA was helical, meaning like a sphere, and like a helix, and like the alpha helix, if you recall, from protein. And they were also able to determine how big the helix was and the spacing between the nitrogenous bases. So from this picture, they were able to come up with a ton of information. And so basically, this is the structure of DNA that they came up with from Rosalind Franklin's pictures. And you guys probably recall this. It's double-stranded, and it's basically like a spiral staircase. So there's one strand on either side. The edges of the staircase, or the backbone of the DNA, is made up of sugar phosphate repeating. So it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, the whole way up and the whole way down. And then they're connected in the middle by the steps or the rungs of the ladder, which are the bases. A matching with T, C matching with G, G matching with C, T matching with A, etc., etc. And that's why Chargaff was able to determine that the number of A always matches the number of T, and the number of G always matches the number of C, because they go together. It's a double helix because it's on both sides, and it's a helix because it twists and goes up, looking like a spiral staircase. But the two sides, actually, they don't run the same way. They run anti-parallel to each other. You guys remember this from chapter 4. One strand goes up, one strand goes down. And that's going to be really important, so make sure you remember that. And we use the different sugars on the 5-carbon sugar, or the different carbons on the 5-carbon sugar, to know which way it goes. So right here, we've decided that this carbon is called the 3, and this carbon is called the 5. So this is the 3' end and the 5' end, which is opposite from over here, which over here is the 5' end, and there's the 3' end. So they are actually running in opposite directions. And you'll notice that the rungs are always made up of one pyrimidine and one purine. This one small rung and one fat rung.